Gene editing, to edit or not to edit. As you may know, science has led us to amazing advances in medicine since a worldwide venture called the Human Genome Project that began in 1990 and completed in 2003. Once the DNA structures and components were mapped, scientists and doctors discovered ways to trace many diseases and disorders that are caused by changes in the structure of an individual's DNA. With the continued advancement of science, the technology to edit human DNA is emerging. One of the most promising forms of DNA editing is called CRISPR-Cas9, otherwise known as clustered regularly interspaced short palindromic repeat with the Cas9 protein that seeks out and cuts DNA. This allows insertion or deletion of new DNA. Here's a video by McGovern Institute for Brain Research at MIT.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
There are of course concerns about editing DNA. The concerns include people creating designer babies or super soldiers. It would be possible to choose a certain height, build, eye, and hair color of an embryo before it is implanted. There are concerns over off-target changes to the DNA that can then transfer to offsprings for generations to come. There are moral and ethical concerns about allowing nature to take its course, a survival of the fittest mentality. There are concerns about using the technology for enhancements rather than therapeutic applications. There is also a concern that this would create a larger divide between the haves and the have-nots, that the technology could be used by those with the resources that are not available to all. Now, I'll put a face to the topic of gene editing and the impact it could have on individuals and their families. This little guy is my nephew, Jaden. 15 months ago, Jaden was diagnosed with a fatal disease called Duchenne muscular dystrophy, or DMD for short. DMD is a progressive muscle wasting disease that affects about 1 in 3,500 male births. DMD leaves boys unable to walk by about 12 years of age, and the average life expectancy is in the early 20s. There are new and experimental medications being used to treat boys with DMD right now. About 8 to 13% of DMD patients can benefit from these treatments. Jaden is lucky enough to be in this category. Jaden goes to Stanford Medical Center once a week for an infusion of a medication that enables his body to produce an abbreviated version of the dystrophin protein that his body is lacking. He has completed 52 weeks of treatment. This treatment, however, is not a cure. It will hopefully extend his mobility and his life expectancy. Jaden also goes to Stanford every 6 months so that they can test his ability to walk for 6 minutes and check his ability to get up from lying down, along with other motor and neurological developments or delays. Here is the lead scientist, Dr. Eric Olson, to describe how CRISPR-Cas9 would impact boys with DMD.
4,000 different mutations in the dystrophin gene that cause Duchenne muscular dystrophy. And by correcting those mutations using CRISPR-Cas9 gene editing, the cause of the disease can be eliminated. Exons are short segments of DNA that code for portions of proteins. If there's an exon that has a mutation that blocks the production of the protein, we can use this technology to skip that exon and put the protein back to restore the production of the missing protein, in this case, dystrophin. The mutation responsible for the disease can be eliminated following that single, one-time delivery of the gene editing components. In that way, it's different from all other types of therapies which require lifelong treatment. We've optimized this technique in human cells derived from patients with DMD. So we are moving rapidly towards clinical translation of this technology. We believe we can correct up to 80% of the mutations identified in DMD boys worldwide using CRISPR-Cas9 gene editing.
How would it impact your family if this science could be used to cure DMD?
Personally, I'm going to allow for a cure to help cure my child. Right now, general life expectancy for kids with DMD is into their early 20s. With a cure like CRISPR that could genetically alter that gene, would allow him to have a normal life expectancy and live a full normal life at that point. So it would be huge for us if it was approved.
While it's understandable that there are concerns over the possible misuse of CRISPR-Cas9, the concerns need to be balanced with moving forward. This technological advance could change the lives of hundreds of thousands of individuals and families. These lives could be forever transformed by curing terminal diseases for them and their loved ones. CRISPR-Cas9 could save millions of lives and cure numerous genetic diseases and cancers. One of the co-founders of the CRISPR technology, Dr. Doudna, has called for a worldwide moratorium until the scientific community has carefully considered how this technology should be used and how it could be misused. As a result, this would waste valuable time that researchers could use to perfect the technology and save thousands of lives. This revolutionary scientific discovery should not be withheld from the public due to fear or misguided beliefs.